Sorry, welcome back. I just realised I've accidentally ended the first video, so hopefully there's no issue with you moving straight on to the second video. So in terms of differences you should have spotted, what you should have seen is that whilst DNA has cytosine, guanine, adenine, and thymine, RNA has cytosine, guanine, adenine, and uracil. So instead of thymine, we have uracil. If you look very closely at the structure of thymine and uracil, you can see the differences between them. But in reality, they're extremely similar. The reason it's replaced with uracil is as a distinguishing factor from the DNA. It's one of the things that allows it to leave the nucleus, one of the reasons why it isn't stopped at the nuclear pore. The other differences you could have spotted is that RNA is single-stranded rather than double-stranded. RNA is single-stranded rather than double-stranded, and that means that it's effectively a smaller molecule, but it's half the size of the same size strand of DNA. Now, you can see that in terms of, actually I want to move on to the next slide because I don't think you can actually necessarily see it there, but it's going to be made of ribose rather than deoxyribose. So not very easy to see on the diagram, but deoxyribose is the sugar that's used in DNA, ribose is the sugar that's used in RNA. And the fourth difference, which you couldn't necessarily see in the diagram, is that it is extremely short compared to DNA. Each RNA molecule is only 1 gene long. So you make a specific RNA copy of a very short piece of DNA. You don't copy the whole chromosome. Because in reality, you only ever really want 1 gene at a time. Let's say the gene for eye color, the gene for nostril hair length. You only want one thing at a time. You don't need a whole chromosome. And so RNA strands are much shorter. Now, I'll give you just a minute to copy that down if you're copying it down. Alright, not exactly 1 minute, so I hope that's enough time. If not, obviously just pause me. Moving on, the kind of questions you'll see in an exam paper are very often just compare and contrast the structures of RNA and DNA. So, what I suggest you do is you draw yourself out a little Venn diagram like this, and at the end of this lesson, just sit down and try to, without looking back, write down as many similarities and differences as you can think of. Now, I've gone through the differences there because they're the things that aren't new, but in reality, there are huge numbers of similarities. So, spend some time after I've finished talking, just going back, trying to do from memory similarities and differences, because in reality, when you come to an exam question, you're going to find that this is what you're going to be asked about. Though probably not in Venn diagram form, often in a table or even just a 2-mark, give one similarity and one differences between the structure of RNA and DNA. Okay, I'll move on from there and you can do that later yourself. Again, these questions you can do yourself. So the next bit we need to cover together is this. Now, if that is a strand of DNA, then you have to think about what the RNA strand would be that RNA polymerase would make. Now, I'll give you 40 seconds, just have a look at it. What would you think would be the sequence of bases in the RNA strand, given that that is the DNA strand, the blue one at the top? Just have 40 seconds to have a look at it. Right. So we'll go through that in a second. One thing I do want to say just before I move on and give you that answer is that if there are any issues with this loom, if you think that perhaps I'm going too fast, I'm not giving enough time for the tasks, I do want to hear about it on Teams. Now, this is the first one I've made, so I actually would very much appreciate some feedback. You've got A, T, G, 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 etc. there. When you're trying to work out what the RNA strand would be, which RNA polymerase would make, it needs to be complementary. And that means, traditionally, A with T, C with G. What we have to remember, of course, is that every thymine, every T, is replaced with a uracil, a U. So if you're going to put in a T, then it has to be a U instead. So the first one, what would go opposite that A would be a U. Then we would have an A, 4 C's, a G, an A G, a U, a C, a G, a C, a U, and a G. So the first strand should be UA, C, 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 GA, GU, CG, CU, G. So it's complementary, but if you would think there'd be a thymine, it's going to replace with a uracil because this is RNA that we're making. This is an incredibly common question. So have a go at another one. Now, if you can't read that, it looks a bit blurry on my screen, and it's just the way it's been stretched. You can write out these letters if you can't read it. G, C, C, A, T, 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 C, G, A, T is DNA strand. What would the RNA strand be? Again, I'll give you a minute just to do it. Right, a minute never seems longer than when you're staring at yourself in the bottom corner of a screen. Now, the answer to that one should then be, again, complementary C, G, G, U, A, G, C, U, A. Fine. Excellent. Now, moving on then. If you can do that, that's the second most common question after comparing and contrasting structures of RNA and DNA. Now, there's one more little fact we have to know. Now, I have to say we never talk about this again. It's just one thing, a 1 mark question you could be asked. In fact, it was the first question on the A level the first year I moved here about 5 years ago. So it does come up occasionally. That fact is simply that ribosomes are made out of a mixture of protein and RNA. Now, you may have been taught that as part of your cell biology course, but sometimes you're not. If you don't know that, ribosomes are made of protein and RNA, it's just a fact you have to memorize. It's not a difficult fact. Just memorize protein and RNA. So that particular picture I've picked there makes it look a bit like the large subunit might be the protein and the small subunit the RNA. In actual fact, they're each made of a mixture of both, so don't get confused by that. Protein and RNA. Straightforward. Nice and easy. Hopefully you've all done the notes. Moving on. I think we're coming straight to some exam questions now because that's about it. Yeah. So I'm going to leave that on the board for 1.5 minutes, but I don't think it will take you that long, but it's a 2-mark question, so you should have some time for it. I'd like the names of D, E, and F. And that is a DNA molecule, not an RNA molecule. Alright, that's a minute and a half. Now, if you find these tasks are taking you too long, I don't think this one will be one of them, but if you do, you can just pause me. If you find that if you're finding that they are taking too little time and you're finding you're just staring at me, staring at you, then do just skip me forward. There's no point, I suppose, sitting in silence. So here we've got D, phosphate, E, deoxyribose sugar, F, base, or you could say nitrogenous base if you want to be really specific, that would be wonderful. With E, it does have to be deoxyribose sugar. If you're a GCSE student, you can get away with just sugar. With E, we're going to be more specific just because, obviously, RNA doesn't contain deoxyribose, it contains ribose, so it's a distinguishing factor. So, phosphate, deoxyribose sugar, and base. And here is the classic question I told you about. Now, it's not a Venn diagram, this one. This one is a table, complete table to give two differences between the structures of DNA and the structure of RNA. Now, that should be, I'd hope, quite straightforward. 2 marks, 2 minutes. Alright, that's 2.5 minutes. Was it? It was enough time, regardless. Anyhow, the answers. So, what I would be careful with this question, with any question, this is the structure of DNA and the structure of RNA. So to make sure you're talking about the physical structure, you can't be talking about their uses or where they're found, just the structure. So, if we move down, again, it's told you D, E, and F, it's been given that. Now, DNA double-stranded, RNA single-stranded, DNA deoxyribose, RNA ribose, thymine versus uracil, very long, very short. You've got double helix for DNA and no double helix for RNA, longer or shorter, but it has to be a comparison. It can't just be a fact. Now, you'll notice that list rule applies at the bottom. I want to mention that just because it comes up a lot when I'm marking questions for your exams. That means that if you wrote more than 2 things, the first 2 things will be the ones that are marked. So if you wrote 2 things per box, only the first 2 things you've written will be marked. Now in a box like that, it's very obvious. If you see a box like that, it's obvious you have to write a fact in each one. I often see when people are asked to write 2 differences between the structure of DNA and the structure of RNA, people are writing 4 differences. That means if one of the first 2 happens to be wrong, or not on the mark scheme, let's say you picked that one was found in the nucleus, one wasn't, you will not get 2 marks, even if the other 3 are correct. So when it says to give 2 differences, please do give 2 differences. Easy in this table, a little bit more tempting to give more in a written format. Fine. Now, that's the end of DNA and RNA. In terms of what you should know, let's have a look at the learning objectives. You should know that it's found in all cells. You should know that it transfers information from DNA to ribosomes. You should know that it's formed of nucleotides. You should know what a phosphodiester bond is already from the previous lesson. You should know that it's formed from ribose, base, and phosphate, that it's short, and that the ribosomes are formed from RNA and proteins. If there's any confusion there, and there's no issue if there is, I'd like you to contact me via Teams and just ask me the question, what is the issue you're having? Because it's very difficult with you obviously being here for me to know what you might be struggling with. Hopefully that's all right. Now, it seems that I've done about 25 minutes worth of video, probably indicates I've spoken far too fast. I suspect if you're watching this on YouTube, you can probably half speed me. After all that, I would make sure, if I were you, that you're completing lots of questions on this. And the temptation when you're given a live lesson like this is just to listen to me talking, perhaps with an Xbox on in one corner, and not to actually do any work. If you're just listening to me talking and not doing any work, you will not have understood this topic, or at the very least, you will forget it in 2 weeks. You have to be doing questions and tasks. And I'll be setting some exam questions with this on the Teams lesson I'll be sending out. Please do complete this. After all that, I hope that's been very useful to you. Looking forward to seeing you all back in school whenever you happen to get back here, hopefully next Friday. And I will speak to you all soon.